everyone and welcome to my YouTube channel. In today's lecture, we will be discussing about the primer designing and the essential tools and softwares we can use for designing a primer. Now the question arises why designing a primer is so important? Because it is a preliminary step in many techniques like polymerase chain reaction and also in cloning. So to make sure that the polymerase chain reaction and the cloning processes takes out to be very efficient, it is very necessary to design a perfect primer. Okay, so first of all, what is polymerase chain reaction? So polymerase chain reaction is a revolutionary technique in molecular biology that allows for the amplification of a specific DNA segment. You guys must have read about DNA replication that occurs in all the organisms. Okay, so what happens there? In vivo conditions, we have an enzyme called primase, which provides the primers, the RNA primers. Using these primers, the DNA polymerase can synthesize a new strand against a template DNA strand. That is what replication is, and that is how it takes place in vivo. But in vitro conditions, we don't have such enzyme called primase, which will provide us with the primers, with the short nucleotide sequences, which are important for the DNA polymerase to synthesize a new DNA strand. So, if we want to synthesize a new DNA strand against a template DNA, or a complementary DNA or a specific DNA segment, it is important that we will provide short DNA segments so that the DNA polymerase can use these segments and can synthesize a new DNA strand against a template DNA strand or the gene of interest. Okay, so polymerase chain reaction is that kind of process in which using these primers, the DNA polymerase can amplify a specific DNA segment multiple times. Okay, and can lead to a million or billion copies of that specific DNA segment. So basically, it is the amplification of the DNA segment of interest. So this process enables researchers to create millions of copies of DNA sequences, facilitating various applications in genetics, forensics, and medical diagnostics. Right? In genetics, especially cloning, we do. So polymerase chain reaction is a very preliminary step or important step in the cloning process. So this is how it takes place. We have a template DNA sequence. Okay, we have this template DNA, which is a double-stranded DNA sequence. It could be anything. It could be plasmid or a genomic DNA. Okay, so the next step, what happens? The first step here is the denaturation. So in denaturation, what happens? This double-stranded DNA gets separated into the single-stranded DNA. So to do so, we require a higher temperature around 94 to 98 degrees Celsius. So this temperature will lead to the disruption of the hydrogen bonds, and so the two strands get separated. So this is the first step in the DNA polymerase reaction. The next step is the annealing, in which what happens, the primers get annealed to a specific sequence. This is the target sequence or against which the primers were formed. So the primers are going to bind to its complementary DNA sequence on the template strand. So this is what we called primer annealing. This is carried out at a temperature around 50 to 65 degrees Celsius, and this totally depends on the melting temperature of the primers that we have designed. We will be discussing about these terms like melting temperature and annealing temperature. What are these terms? We will be discussing it later on. But to understand the whole these steps which are occurring in the PCR, you should get to know about these things. Okay, so the second step is the annealing step, which take place at the 50 to 65 degrees Celsius, a lower temperature basically, where the the primers get annealed or get bind to the complementary DNA sequences. Okay. After that, the DNA polymerase will use these primers and synthesize a new DNA strand against this template DNA strands by adding the nucleotides to the 3' end of the primers. So this is how the polymerase chain reaction take place. This whole cycle will be repeated multiple times, and it will ultimately provide us with multiple copies of desirable gene sequences. Okay, so now moving on to what primers basically are, we'll be understanding the primers. Okay, so primers basically are the short single-stranded nucleic acid sequences which are used by the DNA polymerase enzyme to initiate the DNA synthesis. So if we talk about the polymerase chain reaction, in that we require two primers. First one is the forward primer and second one is the reverse primer, and both the primers are prepared in the 5' to 3' direction. Okay, so one primer, that is the forward primer, is required for one strand, and the reverse primer is required for the another strand, so that in both the directions the PCR amplification will take place, and ultimately we will get the double-stranded DNA as a result, or 2 copies and then 4 copies and then 8 copies, and that is how a million copies of DNA will be synthesized. Okay, now the design of the primer is very critical to ensure the specificity and efficiency of the PCR amplification. Why? Because the the primer which we are like preparing or designing, if it is not complementary with the template sequence, okay, if it is not specific for the target gene sequence, this primer is going to bind anywhere, okay, non-specifically, and that is what we don't want. We want it to specifically bind to the target sequence only, and that is why we will keep that sequence in mind while designing the primers, so that our primers will be specific and more efficient. Okay, so that is what we have to keep in our mind while designing these primers, that these primers should be very, very specific for the target gene sequences which we want to amplify, right? Okay, now there are certain key factors that has to be kept in mind before we design the primers. The first one is the length of the primer. So the length of the primer should be in between 18 to 30 base pairs, depending upon what kind of template you are using. So if you are using suppose the plasmid DNA as a template, so you can use like approx 25 to like 20 to 25 base pair long primers you can use. But if you are using genomic DNA, which is quite larger in number, okay, so at that time you can increase the base pairs of your primers. To maintain a particular length of the primer is very, very important. So depending upon the template which you are using, you can change the length of the primer. Okay, so if you will prepare that or design that primer very short, if the length of the primer is too short, non-specific binding will take place. The primer will bind anywhere non-specifically, and you will not get the proper results. Similarly, if your primer is very long, there will be slow hybridization. Okay, so maintaining a proper length of the primer is very important. Next key factor that we have to keep in mind is the GC content. So the GC content should be between 40 to 60%. It should not be very low or it should not be very high. It should be in the range of like 40 to 60%, with the 3' end having GC clamp to promote binding. So the 3' end of the primer should have the GC content, more GC content, so that the primer will properly bind to a specific region. Okay. Now, what happens if the GC content is very low? It can, if the GC content of your primer is very low, the denaturation of the DNA will be very easy. Okay, and that this is ultimately or indirectly we can say it's going to affect the efficiency of the polymerase chain reaction, because the DNA will get denatured very easily. So we don't want our PCR amplification efficiency to be like degraded or deficient. So to maintain a proper GC content, we can increase the length of the primer in that case, so that our GC content will be increased. Similarly, if we have very high GC content, that may lead to mismatches, or it may lead to the formation of primer dimers or the secondary structures or the hairpin loop like structures. If these structures will form, the primer dimers or the hairpin loop like structures will form, the primers will not going to bind specifically to the complementary DNA sequence. It will dimerize with itself, okay, and that will be useless. So maintaining a proper GC content is also very important. That should not be very low and that should not be very high. We can calculate the GC content of our primer using a very simple formula in which you should know the G's and the C's present in your primer and the whole count of your nucleotides. Okay, so the number of G's plus the number of C's divided by the total number of the base pairs into 100, because we are calculating the percentage. So for example, we have this primer sequence here. So I have highlighted the G's and C's with red and the T's and A's with black color. So we'll put the number of G's and C's in the formula along with the total number of nucleotides, and ultimately we'll get the result, that is 46.66%, approximately 47%. So this is an ideal GC content that can be used in preparation or designing the primer, okay, which is in the range of 40 to 60%. So this is the ideal GC content of your primer. We will be discussing some softwares we can use to calculate this GC content when we'll be discussing about the melting temperature in the next slide. So they will be covering the softwares we can use for this GC content. So manually you can use this formula to calculate the GC content. Next key factor we have is the melting temperature. So what is the melting temperature first of all? So melting temperature, in a very simple term, it is that temperature at which half of the primer is bound to the complementary sequence and half of it is not. So this is a very lame kind of language I'm using to make you understand about the melting temperature. The temperature at which half of the primer is bound to the template DNA and half of it is not. So the melting temperature, it is the temperature at which half of the primer and its complementary sequence are present as a duplex, or it is the temperature at which one half of the DNA duplex will dissociate to become single stranded. Okay, so this is how we can explain the melting temperature of the primer. Now the melting temperature of the primer should range between 52 to 75 degrees Celsius. If your GC content is high, ultimately it is going to affect the melting temperature of the primer. If the GC content is high, melting temperature will be high. If the GC content is low, melting temperature will be low. So the melting temperature is directly dependent upon the GC content of your primer. So design your primer in such a way that the melting temperature should not exceed a particular range. Okay, because it's ultimately going to affect the efficiency of your PCR if these factors are not in the optimum conditions or optimum ranges. Now the primer with melting temperature in the range of 52 to 58 degrees Celsius considered to be ideal as it provides the best results. Okay. Now, second thing that you have to keep in mind before preparing or designing your primer is the melting temperature of the primers, the forward primer and your reverse primer. So forward primer and reverse primer, the difference between the melting temperature of these two should not exceed 5 degrees Celsius. It should be between 5 degrees Celsius. Why I am saying this? Because in the starting of this video, I have explained you different temperature gradients we use in the PCR amplification. Okay, a particular range of temperature we can use. Okay, so while the annealing temperature comes, if the melting temperature of your primer is very different from each other, and it is not within the range of 5 degrees Celsius or so, and the difference is very high, so during the annealing, the primer will not bind to the target sequence or the complementary sequence efficiently because of the temperature difference, and that is why the efficiency of the PCR will be like inhibited, or you can say it will be affected. We don't want that. So to avoid it, we should maintain the temperature of both forward and the reverse primer in between this 5 degrees. So the temperature range should be between 5 degrees. It should not be very high because it's ultimately going to affect the annealing temperature and the whole PCR reaction. So this is the formula we can use to calculate the melting temperature of a primer. So melting temperature is equals to 4 degrees Celsius GC content plus 2 degrees Celsius AT content. So this is the example. This is a sequence of a primer in which similarly I have highlighted the G's and C's with red and T's and A's with black color. Okay, we'll be counting these G's and C's and we'll be putting the number of these G's and C's in the formula, and similarly we'll put the number of A's and T's in this formula and we'll be calculating it as per the formula, and ultimately we'll get the result that is 72 degrees Celsius, which is very ideal melting temperature for a primer. Okay, now there are various softwares or tools you can use to calculate the melting temperature. First one is the oligo evaluator from Sigma Aldrich. Then you have TM calculator from Thermo Fisher Scientific, and then you have oligoanalyzer TM tool, IDT, integrated DNA technologies. So you can use any of these tools to calculate the melting temperature as well as the GC content of your primers. So this is a kind of screen you will get when you will open the oligo evaluator from the Sigma Aldrich. I've given an example here so that you will be able to understand it more specifically and clearly. Okay, so this is the kind of screen you will get at the oligo evaluator of the Sigma Aldrich. Here in this box you will be typing or you will be copy and pasting your sequence, your primer sequence. Okay, so here we have pasted our primer sequence, and then we'll go to calculate. When we'll calculate it, it will show us this kind of window. Here you can see that it has calculated the melting temperature of the primer as well as your GC content. Okay, it will also tell you whether it's forming any kind of secondary structure. So very weak kind of secondary structures are forming. Also the primer dimers are very low, or we can say there is no primer dimer formation is there. So this is a very ideal kind of primer which we have designed. Okay, so you can also design such kind of primer, and you can also calculate the GC content and the melting temperature of your primer using these tools, or you can also calculate it manually. Okay, so this is just one example of the software which we can use to calculate these two factors. Next in line, we have the annealing temperature. So this is the temperature at which the primers are going to bind or anneal to the target or complementary DNA sequence for the amplification, right? The annealing temperature directly depends upon the melting temperature of the primers. So if the melting temperature of the primer is high, you can use higher annealing temperature, and if the melting temperature of the primer is low, you will be using low annealing temperature because it is the temperature at which your primer is going to bind to the DNA sequence. So it's totally depend upon the melting temperature of your primers. The ideal annealing temperature ranges from 55 degrees Celsius to 70 degrees Celsius. These are the ideal ranges of the annealing temperature. So you have to keep all these factors in your mind while preparing your primers. Now there are certain notes I have prepared for you that you have to keep in mind while preparing your while designing your primers. First of all, there should not be any complementary regions in between the primers so that you can avoid the primer dimer formation or the hairpin loop structures in the secondary structures. Here you can see the primer dimers are forming. Okay, so you have to avoid these and these kind of structures, because if primer dimers will form and if the hairpin loop like structures or the secondary structures will form, the primers are not going to bind efficiently to the target sequence or the complementary DNA sequence which you need to amplify. Okay, so to avoid that, you should not be having any complementary regions in between the primers. Okay, then if you are preparing the primer for cloning purposes, in such cases, we add some restriction site upstream of the primers at the 5' end. So suppose this is your primer sequence you have prepared. Okay, now you have a cloning site, like suppose this is the site of zoo. So you have a vector in which you have to insert a specific gene sequence, okay, at the zoo site, zoo restriction site. So what will you do? You will prepare a primer which is specific for your targeted gene sequence, and at the 5' region upstream to this primer you will add the restriction site for zoo. This is the restriction site for zoo 1. Okay, now if you are adding this restriction site, it is mandatory to add 3 to 6 base pair clamp upstream to this restriction site so that the enzyme can cleave the restriction site efficiently. If these base pairs are not there, the enzyme will not get the space to sit there and cleave the restriction site to. So in order to let the enzyme cleave this restriction site efficiently, you have to add some 3 to 6 base pair clamp upstream to the restriction site. So these are the points you need to keep in mind before designing any kind of primer. Okay, so that's all with today's lecture. If you have any query related to the primer designing or something, you can comment in the comment section. Maybe I'll prepare some more videos on the primer designing in which we'll be like discussing about some softwares we can use for designing the primers, like primer 3 we can use, how we can use that software. Also we'll be discussing about how we can use snapgene and many other softwares for primer designing. So till then, if you have any query related to this much part of the primer designing, you can ask me in the comment section and I'll be happy to help. Thank you so much. Bye bye.